This presentation is on the cell cycle and mitosis. Our key concept is cells divide during cell mitosis. It's important to remember that it is the genetic material that must be transferred from one cell to a new cell in order for that new cell to work properly. So it's important to understand that chromosomes are essentially the DNA that are wrapped up in proteins. And you can see from these diagrams how the DNA is wrapped up in essentially what are called histones. And then those histones wrap upon themselves and become these supercoiled DNA strands, which ultimately become what this photograph looks like, which is the sort of X-shaped, if you will, chromosome. One thing that'll come up in this discussion is that the genetic material sometimes has different names, even though it really is the same stuff. So the chromatin is the DNA plus the proteins. When a chromosome has already replicated or copied itself, one of those is called a chromosome, but one half of that is referred to as a chromatid, or if you will, a copy is called a chromatid. So in this picture, you actually see two chromatids held together at what's called the centromere. As a whole, this structure is called a chromosome. Mitosis and cytokinesis essentially produce two genetically identical daughter cells. Typically, the cell starts out in interphase, and it's during interphase that the cell spends most of its time, which is the kind of living phase, if you will, of cells. And then at a certain point, the chromosomes will be replicated when it's time for the cell to divide. Again, remember that each cell needs to have the appropriate amount and number of chromosomes as the original cell. Mitosis then begins essentially in four phases. During prophase, the chromosomes will condense and become these X-shaped objects, again, having each of them being held together at a centromere and having individual chromatids, two individual chromatids for each one. This diagram shows on the left a diagram of the cell in prophase. On the right, it's trying to show an electron microscope picture of the cell in prophase. The dark blue are the condensed chromosomes. The green is the spindle fibers. And in this photograph, you can't see the cell membrane. The next stage is metaphase, typically referred to as metaphase because meta means middle. This is not only the middle phase of mitosis, but also the chromosomes line up at the center or the equator of the cell, or middle of the cell. And as you notice, they are now being attached by the spindle fibers at the centromeres. The next phase is when the cell begins to pull apart the individual chromatids towards the poles of the cell. This is referred to as anaphase, and this is where sister chromatids will separate into opposite sides of the cell. The last technical part of mitosis is telophase, where the cell begins to reform into the two new cells. And again, in this diagram, you'll notice that there are the same number of chromosomes that was original to each cell, in this case 4, and each color is the same. That's indicating they each have the same kind of chromosomes as well. Lastly, there is what's referred to as cytokinesis. This process is basically the process where all of the other parts of the cell are put back in place and put to work so that the new cells can actually work. Those are the steps of mitosis. Essentially, you have interphase, prophase, metaphase, anaphase, and telophase in order. It's important for cells to regulate their growth. Otherwise, we have some issues in an organism or body. So there are several things that can cause different problems, but external factors such as drugs, carcinogens, and radiation can cause the cells to divide either too slowly or too quickly. Internal factors like genes and protein control can cause cells to also grow at wrong or different rates. Apoptosis is the programmed cell death. That is basically the process where cells actually die. An example of this would be where the body's skin cells are programmed to die once they reach a certain level of your skin, the outermost level of your skin, and they actually form a protective layer of dead skin cells. In other places, that occurs as well. Some scientists believe that actually your ultimate death occurs when your body cannot produce any more cells, or the important cells at least, through mitosis. One place where cell regulation is really important and becomes a problem is in the process of formation of tumors. Cancer cells essentially form tumors. And a cell that's cancerous is a cell that's growing out of control. As you see in the diagram, you can see cancer cells beginning to replicate out of control. If the cells stay within a boundary in an area and can be easily removed, we refer to them as benign tumors. If they metastasize or break away or flood into the bloodstream, as the diagram is trying to show you, and then spread to other parts of the body, that's referred to as malignant. And that's the type of cancer that typically is very damaging or can kill you. So cancer cells do not carry out necessary functions, and cancer cells do come from normal cells which have been damaged in some form or another, again through potentially radiation or chemicals or things like that. The type of substances that promote cancer are standardly referred to as carcinogens. Standard cancer treatments typically kill both cancerous and healthy cells, and that's why chemotherapy is so bad, because it often will kill off the normal cells along with cancer cells. This is why someone who has undergone chemotherapy often has no hair, because most chemotherapies affect fast growing cells. Cancer cells are fast growing, and so hair cells, as an example, are fast growing as well. So the hair cells begin to fall out or die because the cancer treatment is killing both types of cells.